Hello everybody, today we start with the next chapter in bio, that is chapter number 6 of plant physiology, that is seeds, structure and germination. So in the earlier chapter we had learned about the different kinds of pollination, different types of pollination and different different processes what takes place during pollination and fertilization. So we understand that a flower, when it is going to be pollinated, it is going to go for fertilization of the egg cell which is present in the ovule, and one that is there, the seed is ready. So that means a zygote is ready with inside that particular fruit, inside that particular flower के नीचे का ovary का part, and then the fruit is going to develop around it. So the seed is nothing but a baby plant or the zygote which is going to be encapsulated in a particular covering so that it is protected from the environment. So the seed, so when we see that seed, so what is a seed? So what is a seed? Before we talk about the germination of seed, it would be appropriate to refresh our knowledge about the actual meaning of the terms fruit, seed and grain. So yeah, we always we get mixed around with the fruit, seed and grain. So let's understand what is the difference between a fruit, a seed and a grain. A fruit is an enlarged, ripened ovary, the ovarian wall forming the fruit wall which encloses the seeds. The fruit protects the seed and helps in seed dispersal. For example, mango or pea pod. So you can see the fruit is basically the one in which the ovarian wall are going to be you can say enlarged and ripened, and you are going to form the fruit wall, and enclosed inside will be the seed. So the fruit is the protective mechanism, or you can say the fruit protects the seed which is inside and also helps in the seed dispersal. So example over here will be the mango. So the outer covering of the fruit is the fruit wall and the seed is inside that particular. So there is a fleshy covering to protect the seed inside. Whereas the seed is a ripened ovule. When the ovule of that flower has ripened, it has got matured, then what is formed is the seed. It contains an embryo, that is a baby plant, which develops into a new plant. The seed coat protects the embryo from mechanical damage, like an example of bean seeds and pea seeds. So pea and bean seeds. You can see that the seed which is going to be having an embryo inside. And then is a grain. Now, what is the difference between them? The grain is the one, it is actually a fruit in which the fruit wall and the seed coat are fused together to form a protective layer, like example maize and wheat. So when the fruit wall and the seed wall, okay, the seed and the fruit wall, they both fuse together, means there is no flashy thing on top of it, then that particular fruit which is there, it becomes a grain. So a grain is a fruit, okay, but in this case there is no differentiation between the fruit wall and the seed wall. The seed coat and the fruit wall are the same, that's why it is forming a grain. That's why it does not have any flashy part which can be had as a fruit. So that's why that's a grain. So it is a fruit which has got a seed coat and the fruit wall fused together. Okay, that's the difference between a fruit, seed and a grain. So fruit and grain are actually the same thing, right? But the fruit is flashy, seed is the grain is not. Whereas the seed is a part inside the fruit. Here also there is a seed inside that particular grain, but the difference is that the grain का जो covering है और the fruit का covering है वो दोनों fuse हो गया है, इसीलिए you don't feel the difference between them. That's why the grain looks like a seed. Okay, the grain itself acts as a seed as such because the protective layer are fused together. More about seed. It is a mature ovule after fertilization. So yes, about the seed, it's a simple thing. It's a matured ovule. Okay, fertilization के बाद का matured ovule is the seed. And what does it contain? It contains a tiny plant, the embryo. Right, the tiny plant is the embryo which it contains. The embryo developed from the fused sperm nucleus and the egg nucleus. So when we studied the pollination part or fertilization part में, जो generative nucleus, that is sperm nucleus, which was getting mixed getting fused with the egg cell. So egg cell और sperm nucleus का जो combination fertilization जो हुआ था, that was the fusion जो हुआ था, that is nothing but the embryo. So the embryo is present inside the seed. Third thing, the embryo consists of a radicle, future root, plumule, a future shoot, and cotyledons or seed leaves, that is future leaves. So you can see that the embryo itself is a baby plant. Why we call it as a baby plant? Because that embryo has got radicle, which is the future root. It has got a plumule, which is a future shoot. And not only that, some have got cotyledons, which are also the future leaves of that particular plant. So the first leaves of the plant are the cotyledons. So that is why you find that all these three things are present inside the seed. The embryo remains in an inactive dormant state, but upon exposure to favorable condition, it germinates. So the embryo which is there, it is not going to start germinating in the moment it is just जब वो mango लिया या कुछ भी लिया तो ऐसा नहीं है कि वो mango में से दूसरा plant grow होने लगेगा. It is going to remain dormant. It is going to dormant in sleeping condition. It is going to remain dormant till the time it does not get the required conditions. Okay, till the time it has not got the conditions favorable for its germination, it is not going to start germinating. So that's why it will be only on exposure to the favorable conditions that the seed will start germinating. The seed may also contain endosperm, which is a food material for the nourishment of the embryo during germination. So many times the seed itself may have certain kind of the nutritive content in it. That nutritive content or the fruit which the food what the plant requires for its growth is available inside, and in such a case that particular area, that particular covering or that particular layer of that particular seed is called as the endosperm. So certain seeds, they do have nutritive content in them which is called as the endosperm, which is a food material which is required for the nourishment of the plant when they are in the germinating stage. The seed can withstand unfavorable conditions of temperature, drought, etc. Some seeds are known to remain dormant even up to 100 years or more. So you can see the dormancy of the seed can be sometimes even endless. You know, as they have mentioned over here, that the dormancy can be as great as 100 years. So what exactly happens is the seed is going to protect itself and going to be waiting for the favorable conditions to come. Till that time it can survive extremities like extreme heat or any unfavorable conditions like temperature, drought, etc. without affecting its endosperm, its zygote which is present inside, the embryo which is inside. So that is how the dormancy can be there for a plant, for a seed, even up to 100 years or more. Now let's see the classification and structure of the seed. So seeds vary in size. Some are so small that they are barely visible to the naked eye. Poppy seed, strawberry seeds. Some are quite large, as in mango and plum. Largest seed are those of coconut and double coconut. The size, shape and structure of the seed of different plants can vary considerably, but the basic structure of most seed is the same. So yes, the seed could be very small, as in case of a strawberry. Strawberry का seed तो हमको मालूम भी नहीं होता कहाँ है. Okay, खाते हैं तो पता भी नहीं चलता कि हम लोग ने seed भी खा लिया साथ में. We don't even know. So that is why strawberry seeds are very, very small. There would be something like a mango which has got a very big seed inside. You know that it's a seed inside, so you cannot even eat it. So that particular part is the seed, so you know it very well. And coconut by itself is a seed. So coconut seeds are the largest one. So largest seed are those of the coconut and the double coconut. The size and the structure of the seed may vary, but the basic structure, or you can say the basic structure of the seed will remain the same. Accordingly, we have got two types of seeds. Okay, there are two types of seeds. And these seeds can be on the basis of the endosperm which are present in it. What is an endosperm? It was the food material. The food material which was present, which is present in the plant, in the seed. If the food material is present, then there is an endosperm, and if it is not present, then it is non-endosperms. So, if it has got food, that is, it has got endosperm, it is called as album albuminous. Okay, it is albuminous kind of a seed which is having endosperm. So it is albuminous. Okay, albuminous means with endosperm. With endosperm means with food material, food nutrition. Okay, that is albuminous. In such seeds, cotyledons are thin and membranous and endosperm persists. Monocot albuminous plants will be cereals, millet, palm. Dicotyledonous albuminous seeds will be poppy, custard apple, etc. So this way albuminous के अंदर भी दो part है, that is monocot and dicot. Monocot will be the one which will be having only one cotyledon, so it is monocot, and other one will be dicot having two cotyledons in it. So albuminous will be the one which has got an endosperm, and they will be having the thin membranous cotyledon, and endosperm will be there. X-albuminous is the next one, that is the X-albuminous. That is non-endospermic. So it is non-endospermic. Okay. So spermic. Okay. It does not contain endosperm. In such seed, the cotyledons store food and become thick and flashy. So in such a case, because there is no endosperm of its own, that's why the cotyledons which are storing it, they are going to be flashy and they are going to store the nutrients in it. So that is the non-endospermic kind. So monocotyledons, again, there will be of two kinds. Monocots, X-albuminous seeds will be less in area. Orchids, Amorphophallus. Dicots में gram, pea, mango, mustard. These are the dicotyledonous kind of albuminous, X-albuminous seeds. So there the cotyledons are there to store the food, whereas in case of albuminous, the endosperm will be there to store the food. So broadly the seeds are of two kinds, monocotyledonous and dicotyledonous. Monocotyledonous seed contain one cotyledon, that is one seed leaf, that is maize, grass, etc. And dicotyledonous seeds contain two cotyledons, like pea, gram, beans, etc. You can see that very well. हम लोग जब सिंग खाते हैं तो सिंग भी हम लोग खाते हैं तो वो दो पार्ट में डिवाइड होता है अपने आप. हम लोग थोड़ा प्रेस करेंगे तो दो पार्ट में डिवाइड हो जाता है. चना भी खाते हैं तो दो पार्ट में इमीडिएटली डिवाइड हो जाता है. So what is that? They are the two cotyledons, right? So whereas if you take corn क्या दो पार्ट में डिवाइड होता है? नहीं होता. So that is monocotyledonous. Okay. So monocotyledonous is only one single thing, like a corn. And dicotyledonous like rajma, gram, pea, तो दो पार्ट में immediately divide हो सकता है. Now we see the structure of a bean seed. So let's see the structure of a bean seed. So in case of a bean seed, so you can see the bean seed is like this, a kidney shape. Okay, and this is too big, this part. Like this, you can have a small like this. But now the outermost part over here is called as the testa, and inner layer inside over here, which is the inner protective covering which is inside, is called as the tegmen. Now the testa and the tegmen are the two seed coats or the protective coatings which are there. The concave part over here, this part over here, which is the concave part, is the hilum. That is the hilum part, and above the hilum you can see a small hole, a small opening, a small dot-like structure. This was the micropyle. So this is the micropyle. If you remember, micropyle was that particular point through which the tube nucleus entered into the ovule. So that is the micropyle over here. In a seed, the micropyle is going to help to absorb water inside the plant. So inside the seed. So micropyle, which is going to be there, and then when it is going to start growing, there will be a radicle coming outside and the plumule going out. So so this is the dicotyledonous one. So because it's dicotyledonous, actually the figure will be something like, when it opens up, the figure will be something like this. So it will be like two cotyledons which are over here. So this two cotyledons will be such that there will be some kind of a leaf-like structure coming here, and there will be a root-like structure coming here. So that is, this part is the radicle, and this is the plumule. So plumule and the radicle are going to be the plumule being the root, plumule being the shoot part, and the radicle being the root part. So that is the cotyledons. So these are the two cotyledons which are here, and so what we have is radicle, or we can also write it as the upper part over here. The upper part which is forming the radicle is the epicotyle. Okay, that's the epicotyle which is on the top part. The plumule leaves which are there, the plumule with two rudimentary leaves, and the testa we already discussed. These are the things, and then there is a hypocotyle which is on the lower part. On the lower part is the hypocotyle. So this is over here, I'll just write it over here, hypocotyle. So hypocotyle enlarges to form the radicle, and the epicotyle enlarges to form the plumule. So epicotyle, epi means above, so one which is going to go above the ground will be epicotyle, which is going to grow and form into the leaf part, and the radicle is the hypocotyle. Hypocotyle is lower area, that's why it is the radicle forming the lower part. So that is the hypocotyle and the radicle, and of course the two cotyledons are over here. These are the two cotyledons. So the figure over here, yes, very important. You need to learn the figure to draw as well as to label. So you need to know the different structures in the bean seed. Now, so let's understand what are bean seeds. There are a number of different kinds of beans, such as broad beans, lima bean, French beans, etc. राजमा है, beans से कितने सारे टाइप के ना, वो French beans, fancy, then rajma is there, so valve is there. So there are so many different kinds of beans which are available in the market. So yeah, but the structure as a whole, the structure remains the same. So but the general structure of these seeds, their seeds is the same. Most are kidney shaped with a convex and a concave side. So convex, there's a convex, and there's the concave side, that is like a kidney. Seed coat consists of testa, the outermost hard brownish covering. It protects the delicate inner part of the seed from injury and from the attack of bacteria, fungi, insects. Tegment is the inner layer lying next to the testa and is also protective in nature. So the testa is the outermost part, the brown color, the hard part, which is the testa, and then inner part there is the tegment. Hard part does not protects the seed from the bacteria and fungus and all, where and the inner part is also for protection, that is the tegment. Hilum is a distinct whitish oval scar on the concave side of the seed. So on the concave side of the seed is the white colored scar. So if you take a rajma का अगर हम लोग ने bean लेंगे तो उसमें एक corner on the concave side there is a white colored का एक marking हो जाता है पूरा इतना सा. So that particular marking is nothing but the hilum. It represents the spot where the ovule, now the seed, are attached to the ovary wall through placenta. So hilum is the place where the ovule was connected to the placenta. So that's why it is that particular point where it was connected to the placenta. A tiny pore called micropyle is situated close to the hilum. It marks the opening through which the pollen tube had entered the ovule. So micropyle over here, it is that point through which the pollen tube had entered into the ovule. Micropyle serves two functions now, but after the seed is being formed, the micropyle is going to give two main functions. One is, when soaked in water, the seeds absorb water mainly through this micropyle and make it available to the embryo for germination. So first thing is that when you take any seed, when you take any bean seeds and you put it in the water, what you find after some few hours, they will become completely soft, so much that they can easily be removed into two cotyledons. So that particular, when the sprouting takes place. So ये क्यों होता है? Because the micropyle which is present over here is allowing the water to enter into the seed. So the entry of the water into the seed is taking place with the help of the micropyle. The second, it provides with the diffusion of respiratory gases for the growing embryo. So once the seed is able to get the conditions favorable for it, and then the seed, that is the embryo which is there inside the seed, is going to start its respiration. Most of the respiration is anaerobic, but whatever respiration takes place, so at that time the diffusion of the gases or the respiratory gases, oxygen का लेना देना जो भी होना है, carbon dioxide निकालना है, तो वो जो काम है, that is going to be done with the help of the micropyle. So micropyle will be doing two purposes. One is the water intake by the seed during germination, and secondly is the respiratory gases ka diffusion during respiration of the embryo. Below the seed coat are two thick cotyledons which contain food for the embryo and protect it. So below this intestine tegment are this particular part, this one and this one. These are the cotyledons. Cotyledons are the food, they are nutrition, okay, the nutrition for the embryo. So they are the cotyledons, the tiny embryo can easily be the food for the embryo and protected. On carefully separating the two cotyledons, the tiny embryo can easily be seen attached to one of the cotyledon. The embryo consists of two parts: the radicle, which later forms the root, and the plumule, which later forms the shoot. The plumule consists of a short stem with a pair of tiny leaves and a growing point between them. So if I take a dicotyledonous seed, now the best example I would take will be of groundnuts. Okay, if I take groundnuts, तो ग्राउंडनट्स को जब भी हम लोग ऐसे खोलते हैं तो एक साइड पे कॉटिलेडन के एक एज पे दोनों कॉटिलेडन खोल दिया, एक कॉटिलेडन पे यू विल फाइंड अ स्मॉल प्रोजेक्टिंग थिंग कमिंग आउट. तो वो जो प्रोजेक्टिंग जो बाहर निकला है, दैट इज नथिंग बट द प्लूम्यूल, दैट इज नथिंग बट द एम्ब्रियो. सो एम्ब्रियो कैन बी सीन व्हेन यू ओपन इट अप ओवर देयर, एंड द एम्ब्रियो consists of two parts: the radicle, which is the root part, which is going to grow into the root part, and the plumule, which is going to be the shoot part. The plumule consists of short stem with a pair of tiny leaves, a short stem, which is nothing but the epicotyle, and the tiny leaves which are there called as a plumule. So tiny leaves and a growing point between them, that is the epicotyle. Now let's remember the radicle is CLE in this case, not CAL. CAL is in chemistry. Okay, this is CLE. The region of the axis between the point of attachment of the cotyledon and the plumule is called as the epicotyle, and the region of axis between the cotyledon is called as the hypocotyle. So ये region जहाँ से cotyledon के साथ में connected है, उसके ऊपर वाला part which is going into the plumule part, which is called as the epicotyle. The one which is going to form in the enlargement of the epicotyle will form the shoot part, and the enlargement of the hypocotyle, which is the lower part, will form as the radicle of the root. So the region of the axis between the point of attachment of the cotyledon and the plumule, cotyledon and the plumule के बीच में का part is epicotyle, and region below the epicotyle is the region below the axis of the cotyledon is the hypocotyle. So that was about the structure of the bean seed. So this was a bean seed, which was a dicotyledonous seed. Okay, so this was the structure of the bean seed. Now we see the next structure, and that is of a maize seed. So now we see a maize seed. A maize seed or a corn seed, both are similar. So that's basically a seed which is a monocotyledonous one. So it will be, the seed will be something like this. So it will be just like a, if you take a corn, it will be just like this with a small connecting things over here. And over here you will be having a small dot which will be the remains of the style. Okay, then you have got the plumule position. Okay, I will not say this diagram is not giving you much of a detail, because there is a over here like this and like this over here, which will be, this will be forming the plumule, and this is forming the radicle. Okay, and this is the outer part, which is the endosperm. The endosperm is over here. Okay, there's a fused endosperm. But mainly you will be having this particular figure, which is more important to learn and to know, and that is the figure which is like this. Okay, so you can see over here, the first one is the fused pericarp and testa. So first part over here is the pericarp. You can see the fruit wall, the pericarp and the testa are fused together because that's a grain. So you can see the maize is a grain, and because it's a grain, the fruit wall and the seed coat are fused together over here. And next one, the one is a protective layer, is called as the oleron. So oleron layer is the other protective layer over here, that is the oleron, which is also the endosperm rich, the fruit rich or the rich in nutrients, the proteins. That's the oleron layer which is on the over here. The next what we are having is the endosperm and the epithelium. So this is the epithelium, the outermost part is the epithelium. Okay, and this one is the endosperm. Okay, endosperm contains the nutrients for the plant, for the embryo, and then this is the, so this is the cotyledon. So this is the cotyledon, or the, in case of a monocotyledonous like this, it is also called as the scutellum. And then you see over here that this area over here is going to form the plumule, and this one over here is forming the radicle. When to protect the plumule is the upper layer, which is called as the coleoptile. So this is the coleoptile, and this is the colorizer. So coleoptile and the colorizer are the two layers over here for the plumule and the radicle, and this combinedly is the embryo. So combinedly is the embryo. This combined thing is the embryo, which is out here. So this is the longitudinal section of the maize grain. So see, if you read out the, see the details. The maize grain is actually a single seeded fruit in which the fruit wall and the seed coat are fused together to form a protective layer. Therefore, it is such a fruit called a grain. So this is a grain because there is no flashy fruit part. It is the fruit part, the fruit wall and the seed coat are fused together over here. That's why it is called as a grain. One side of the grain occurs a small light colored oval scar which marks the location of the embryo inside. So this particular part over here is the light colored part, which is the one which has got the embryo inside. The remaining major part of the grain consists of large endosperm which is rich in starch. So this part, major part over here, is the endosperm which is rich in starch. The endosperm and the embryonic part are separated from each other by a thin epithelial layer. So there is a thin layer, epithelial layer, a thin wall layer is there over here, which is the epithelial layer, which is separating the endosperm part with the embryo part. The outermost layer of the endosperm is rich in protein and is called as the oleron layer. So oleron layer is the outermost part which is rich in protein. So yeah, that's why we say that the grains are good for health, for proteins, because grains say oleron layer hota hai, which is good in proteins. The embryo consists of single cotyledon here called as the scutellum, a radicle and a plumule. So this embryo contains over here single cotyledon called as the scutellum, and it has got the radicle and the plumule. The radicle is towards the pointed end and it is enclosed by a protective sheath, the coryza. So the radicle which is over here is covered with a protective sheet which is called as the coryza, whereas the plumule is going to be the upper part which is going to be acting as the plumule is going to be covered with a coating which is called as coleoptile. So the plumule is towards the upper broader side of the embryonic region and is enclosed in a protective sheath, the coleoptile. The maize grain is monocotyledonous and endospermic. Some other example of this type are rice, wheat and oats. So the major difference between the bean seed and the maize seed are: bean seed 2 cotyledons, maize seed 1 cotyledon, scutellum. Bean seed no endosperm, maize seed large endosperm present. Bean seed large embryo, maize small embryo. Bean plumule leaves folded, whereas maize plumule leaves rolled. Beans plumule large, maize plumule very small. Beans hilum and micropyle visible, maize hilum and micropyle not visible. And beans seed separately contained in the fruit called pod, whereas maize the seed walls and the fruit wall fuse to form a single grain with no separate seed. So this was the major difference between the monocotyledons and dicotyledons examples, that is bean seeds and maize seeds. So this was about the seed part of the chapter. So we see the structure of the seed. We have seen the structure of the seed. Now we see the germination of the seed, which I think we'll do it in the next video.